Welcome to Biology with Medmelo. And today we are going to discuss about DNA, its structure, replication, and the genetic significance. So let us know the story of DNA. First of all, let's talk about the structure of DNA. Here you can see that there are two coils which are stranded towards each other as compared to the RNA. That is, it is a double-stranded structure. And since it is made up of many small things, this structure is known as a polynucleotide. Hence, we can say that it is a double-stranded polynucleotide which is rounded in a double helix fashion. And this double helix fashion has two coils which are running towards each other in an anti-parallel fashion. Now, what does this anti-parallel means? That is, if one's direction is from 5 prime to 3 prime, the other would be from 3 prime to 5 prime. That is opposite of each other. Now look at here. This is the one turn of our DNA, as I have drawn over here. This one turn, according to the biological term, is termed as one pitch of the DNA, which is made up of 10 base pairs and is equal to the height of 3.5 nanometer or 34 into 10 to the power minus 9 nanometers. Okay. If we are talking about the human diploid cell, that is double the cell, then it would be 2 times, which means this 10 base pair would be now of 20 base pairs and its height would be 6.8 nanometer. So this was about the structure of DNA, but how the structure is made, let us consider over here. As we've earlier mentioned that DNA is a double-stranded polynucleotide. Now what is a polynucleotide? Polynucleotide is made up of two words, poly and nucleotide. Poly means more than two, while nucleotide is a certain monomer type unit of the DNA. Okay, so when many and many nucleotides compile with each other to form one sequence or a chain, that is considered as a polynucleotide. Now let us outshine some light on the nucleotide. The nucleotide is basically the cluster of three things, that is sugar, nitrogenous base, and phosphate group. You can consider them as three friends studying in the same group, same college, and residing in the same room of a same hostel. Okay. Now there's a twist in the story that sugar and nitrogenous base are best friends since their childhood. These are childhood friends. Hence, they make up a certain type of compound that is nucleoside. And since they're childhood friends, their bond is known as N-glycosidic bond. Now they have a roommate, a friend, and a groupmate that is phosphate group. This phosphate group experiences a phosphodiester bond with them. Phosphoester bond with them. And when this nucleoside bond makes up the group with the phosphate group, they together make nucleotide. Hence, you can say that the nucleotide is made up of nucleoside plus phosphate group, or when bifurcate them, we can say it is made up of sugar, nitrogenous base, and phosphate group. Now let's talk about the sugar which is present in them. The sugar basically we are talking about is pentose sugar and more specifically the ribose sugar. So RNA has the ribose sugar, and since the DNA is two times of RNA, so we can say that it has deoxyribose. If anything which is related to RNA and DNA both, and DNA is the double version or two times version of RNA, just add deoxy in front of that. Remember this point, we are going to use it further. Okay, now we have nitrogenous bases. Now, nitrogenous base is a kind of person in this group which has multi-talents. That is, it has many parts and further bifurcations. It is basically divided to two categories. One, double ring structure, and one, single ring structure. If we are going to talk about the double ring structure, we have purine. This double ring structure is known as purine, which is of two types, adenine and guanine. If we are to remember them, we can use these of the mnemonics, pure as gold and aloo puri gobi. Got it? Now let's come to the pyrimidine. Pyrimidine is a single ring structure, or you can consider it as a pyramid. Pyramid has three sides, and three sides or three corners, which means it has three parts, in which one of them is common in both DNA and RNA, while the other two sides, one is present in DNA while the other is present in only RNA. The one that is common in both DNA and RNA is cytosine. The one which is present in DNA only is thymine, and if we are talking about RNA, at the place of thymine, we would have uracil. Okay, so three corners of a pyramid, that is three parts of a pyramid: cytosine, thymine, and uracil, and you can remember them as crazy Uncle Tom residing in a pyramid. Got it? Now there's a certain role of hydrogen bonds between purine and pyrimidine. Hydrogen bond is basically used to combine them, or we can say to make friends with purine and pyrimidine, because adenine will make double bond with thymine and cytosine will make triple bond with guanine. When we are talking about RNA, adenine will be bonded with uracil. Now, this bond of adenine and thymine is a weaker bond as compared to cytosine and guanine, and we are going to use it in DNA replication. So remember this point also. Okay. Now let's come on to the childhood friendship of sugar and nitrogenous bases. We have learned that sugar and nitrogenous bases form what? Nucleoside. Now, what kind of nitrogenous base makes what kind of nucleoside? It is different in both RNA and DNA, but a single trick which we have learned to remember in the first part, that DNA is what? Two times of RNA. So add deoxy in front of what RNA is made. So we are going to use the same trick over here. If sugar plus adenine are forming a nucleoside, in RNA it would be adenosine, while in DNA it would be deoxyadenosine. Similarly, when with guanine, RNA gynosine and DNA deoxygynosine. With cytosine, RNA cytidine, DNA deoxycytidine. Now, thymine is only present in DNA, so it would be directly deoxythymidine. And since uracil is only present in RNA, it will be uridine. Now, let's talk about DNA replication. DNA replication is a process of making two new daughter cells or replica of the DNA, which are identical to one another by using certain enzymes. This method is semi-conservative method, which means if this is a DNA structure, when it is dividing, each of the parental strand will get attached to the new DNA strand such that the parental gene will be conserved in a halfway, that it is a semi-conserved. This kind of postulate was basically proposed by Watson and Crick during their double helix model, but it was scientifically proved by Michelson and Stahl in 1958, that is 5 years post the double helix model proposition. Remember this point also because we are going to use this further. Okay. Now let's talk about the genetic significance of DNA. The first genetic significance of DNA is genetic information storage. The DNA stores the genetic information in the form of nitrogenous bases, which are adenine, guanine, cytosine, thymine. And these genetic information are then inherited from one generation to another generation. When these characters are transmitted, then the cell would have to perform certain functions which are designated towards this kind of hereditary characteristic and certain metabolism. And if any error occurs, then mutation would be there, and mutation would certainly lead to what? Evolution. So a minor mistake or a minor thing can determine your whole progeny. Got it? Now about this Michelson and Stahl experiment and the Watson-Crick experiment, if you want more information about it, you can tell me in the comment section. I'll make a separate video for that. So this was all about the question. If you have any doubts, please let me know in the comment section. Thank you.